Hi, my name is Naomi Casares, and my protein was deoxyribonucleases. Hope you can see that. Okay. All right, so let's begin. So, what are proteins anyway? Proteins are structurally complex biological molecules, and they serve many functions, including storage, gene regulation, enzymes, structural regulation, transport, transport of small molecules or ions, motive force, and even specialized purposes. They're made up of an amino acid sequence, and they're linked together by polypeptide bonds. So these amino acids will interact with each other in such a way that they will form a functional 3D structure. And with that, they're going to form a cavity, which is also known as the binding site, which allows substrates to attach and eventually allows the protein to catalyze necessary and essential chemical reactions that must occur within the cell for life to occur. So this folding is basically what allows proteins to carry out their functions. And to better understand these complex structures that proteins make up, we think of them in four different levels. So there's primary, secondary, and tertiary. So the primary level just only talks about the amino acid sequence that a protein has. And then we have the secondary protein level, which only talks about the alpha helices and the beta pleated sheets and the irregular folds that are also within a protein. Now, with tertiary, we get that, but everything is involved. So we have the alpha helices, the beta pleated sheet, and the irregular folds all in one functioning protein unit. And then we have the quaternary. And so the quaternary is different because you need several protein subunits for the whole protein to function. So for example, let's take hemoglobin. Hemoglobin is made up of several protein subunits, but if you take only one away, it's just not going to work because it needs all of them to work. So what are deoxyribonucleases? Deoxyribonucleases are enzymes. They cleave DNAs in certain areas, and they're grouped into one of two. So they can either be endoribonucleases or exoribonucleases. And so to narrow down my research, I chose deoxyribonuclease 1 and 2, also known as DNase 1 and 2. And both of these are known as endonucleases because they cleave the DNA from within rather than from the ends of the DNA. And they're mostly found in mammals, but they are found throughout the animal kingdom. DNase 1 will cleave DNA, and that will make it yield 5' phosphomonoesters. And these proteins are mostly found in pancreatic cells and tissues that are involved with digestive processes. And then we have DNase 2. Get to that real quick. And then we have DNase 2, which after catalyzing a chemical reaction in the cell, it's going to yield 5' 3' phosphomonoesters. And excuse me, sorry. I thought I had to sneeze. Okay. So both of these have a similar function in that they both degrade the DNA during apoptosis, which is controlled cell death. And they both serve, overall, they both serve the organism in functioning to help serve growth and development of the organism. So more specifically, I want to talk about DNase-1. So DNase-1 is a glycoprotein, and it's made up of around 258 to 260 amino acids. It has about 2 disulfide bridges in it, and the folding is determined by the residue amino acids at the C-terminus location, position of the amino acid chain. So it requires particular specific cations for this protein to be activated so that it can function. And these cations include calcium, magnesium, and manganese that are positively charged. Okay, and so the optimum pH levels for DNase-1 to be able to function is between 7 and 8, which is relatively neutral. So the tertiary form contains 2 6-stranded anti-parallel beta pleated sheets, and these are attached to 3 alpha helices and other regular folds within the protein. Okay, so here we go. You can look back a little bit. Okay, and so DNase-2 is also a glycoprotein, but it contains glucosamine, and it is made up of 343 amino acids. It only has 1 disulfide bridge. It has 1 activating cation that it needs so that it can be able to function properly, which is sodium. And the optimum pH levels for DNase-2 to function ranges between 4 to 5, which is relatively acidic. And the functioning unit that DNase-1 and 2 have to be in is actually the tertiary protein level of structure. So this one right here is DNase-2, and the colorful one right here is DNase-1. So DNase-1, DNase-2. Okay, so research and genetics. Now that you know what DNase-1 and 2 are, I want you to know that DNase-1 are actually very special in that they were the first nucleases to actually be discovered, and in that they were the first ones to be studied and observed, and they were first found they were first extracted and purified from young cattle rather than being purified and extracted from humans. So it's been located on the human chromosome, and DNase-1 is at chromosome 16, specifically position 16p 13.3, and DNase-2 has also been located on chromosome 19 at position 19p 13.2, and they have both been successfully sequenced. And this is their sequence, so this is DNA-1. And this is DNA-2. So every letter you see here stands for one particular amino acid. So there are some genetic diseases that are involved with DNase-1. And DNase-1 deficiencies is known to cause lupus, which is characterized by frequent inflammation of the skin, and it's accompanied by joint pain and fatigue. And recently, it's been known to be used as a risk factor marker for gastric carcinoma. So doctors can actually look at this and tell if a person is at high risk of later developing gastric carcinoma. And this is some pictures of lupus. And here's the inflammation I was talking about. Okay, so what did we find out about all this? What does this all mean? So the association between proteins and phenotype is very important because proteins affect the cellular function, which ultimately affects the organism's overall health. And therefore, without these proteins, life itself could not be sustained, and replication wouldn't be able to occur. So therefore, following generations would not be able to endure. Thank you.